Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
It's long been a common belief, hasn't it, that once we hit adulthood, our brains become pretty much static, done making new cells, that the neurological hardware is, well, for the most part, set in stone. But what if that whole idea was, completely overturned by new science? Today, we're diving into some truly groundbreaking research that's challenging that very notion. It suggests our brains are far more dynamic and adaptable than we ever really imagined. Our core topic for this deep dive is this fascinating and for a long time really heavily debated question. Can adult human brains still form new neurons? That process scientists call neurogenesis. Our mission today is to unpack some, well, really compelling new evidence. It comes from a recent study published in the prestigious journal Science, and it was also highlighted in a health news article from Medical Express. We're going to dig into the surprising facts, try to really understand the cutting-edge methods these researchers use to uncover them, and explore what this could, well, profoundly mean for the future of brain health, learning, and maybe even our understanding of ourselves.
Yeah, and what's truly fascinating here, I think, is how this study provides such a crucial kind of missing piece to this long-standing puzzle. It's not just confirming things we maybe suspected before, it's actually showing us how new neurons seem to form in the adult brain,
Yeah.
right down to identifying the specific cells involved and even tracing their development. It really solidifies our understanding of brain plasticity in a way we just haven't seen before.
Okay, let's unpack this a bit then. Because for decades, the scientific community has been grappling with this question, right? Adult human neurogenesis, there were always these conflicting views. Some studies suggested it happened, others argued no, it completely stopped after childhood. Can you give us a quick recap of that sort of scientific tug of war?
Absolutely. For a long, long time, the prevailing thought, the dogma almost, was that our brains were pretty much finished developing new neurons after a certain age, maybe early childhood. Then around 2013, things started to shift a bit. There was a significant study led by Jonas Friesen's research group at Karolinska Institute in Sweden. That study famously used this really ingenious method. It involved carbon-14 levels in deoxyribonucleic acid, or DNA. And using that, they showed that some new neurons actually did seem to form in the adult hippocampus.
Right, the hippocampus. And for anyone listening who might not be familiar, could you just briefly explain what that is? Why is it so central here?
Sure. So the hippocampus is this vital brain region. It's shaped a bit like a seahorse, actually, tucked deep within your temporal lobe. It's absolutely essential for learning and forming new memories, particularly, memories related to facts and events. It also plays a role in emotion regulation and even spatial navigation. Finding new neurons there, of all places, was incredibly significant.
Okay, so we had Friesen's 2013 study suggesting neurogenesis in the hippocampus. But you mentioned the debate carried on. What was the core reason for that ongoing skepticism, that disagreement?
Well, it raised a really important question that fueled that debate. If new neurons were forming, where were the precursor cells coming from? The biggest point of contention, the real sticking point, was the lack of clear, direct evidence that the cells that precede new neurons, the ones we call neural progenitor cells, that they actually exist and actively divide in adult humans.
Ah, so like the parent cells, the ones that give birth to the new neurons.
Exactly. Think of them like that. Without definitively identifying these proliferating parent cells, many scientists felt there was still a missing link. You couldn't truly confirm continuous adult neurogenesis without finding the source.
It was like seeing new buildings pop up, but having no idea where the construction workers or the raw materials were coming from.
That's a great analogy. Yeah, that was pretty much the situation.
Okay, so the stage was set. This latest research, again led by Professor Jonas Friesen, clearly had a very specific, very ambitious goal then, to definitively identify these elusive neural progenitor cells and confirm they are indeed driving ongoing new neuron formation in adult human brains. How did they even begin to approach such a complex problem? I mean, what cutting-edge methods did they actually use?
They really pulled out all the stops, technologically speaking. This wasn't just, a simple look under a microscope. It was a deeply sophisticated approach that combined several advanced methods, and they looked at brain tissue from people ranging from birth all the way up to 78 years of age.
Yeah.
They got these precious samples from various international biobanks. Their strategy was really multi-pronged. First, they used a technique called single-nucleus ribonucleic acid sequencing. Now, that's a mouthful, I know. But essentially, it allowed them to analyze the gene activity inside individual cell nuclei.
Like reading the unique instruction manual for every single cell.
Pretty much, one by one, without having to make assumptions about what that cell should be based on older markers. And crucially, this method allowed them to identify very rare cell populations without relying on those preconceived markers. That was a major limitation in previous studies trying to find these new neurons, so it gave them a truly unbiased view of all the cell types present.
Wow. Okay. That sounds incredibly powerful. What other techniques did they layer on top of that?
Well, they also employed something called flow cytometry that helped them further study the properties of these individual cells, things like size and complexity. But then, to make sense of just the sheer volume of data generated by the sequencing, they turned to machine learning algorithms.
Ah, artificial intelligence.
Exactly. These algorithms were meticulously trained. They were taught to identify nuclei that were transcriptionally similar, meaning their gene activity looks similar to the intermediate neural progenitor cells and neuroblasts seen during childhood development. It was a highly specific approach. And they validated its accuracy, too. They tested it in juvenile mouse data sets and even in the adult human cortex, where neurogenesis isn't really expected, found a very low false prediction rate.
So they could identify these specific cell types with high confidence, but finding them is one thing, right? Pinpointing them precisely within the brain tissue, that's another challenge. How did they confirm where these new cells were actually emerging?
That's a critical point. To actually localize these newly formed cells within the tissue, to see them in their natural environment, they used two other sophisticated techniques, one called ribonucleic acid scope, or RNA scope, and another called xenium. These methods basically allow researchers to pinpoint the exact location of specific gene activity within brain slices. It gives them a kind of high-definition map showing where these new cells were emerging, which genes they were switching on.
So you get the spatial context.
Precisely, which is vital. Older techniques, like bulk sequencing, would just give you an overall average, a snapshot, without showing you where the action was happening. That's why these advanced, really cutting-edge methods were so critical for this study's success in proving what was going on.
Okay, so with all that incredible advanced capability, what were the really eye-opening, the groundbreaking things they actually found happening in these adult brains?
Well, this is where it gets really exciting, I think. The study found compelling, and it's a definitive, evidence for the ongoing formation of neurons in adult human brains. And they didn't just find a few hints. They identified all the stages of neuronal development.
All the stages.
Yes, ranging from the initial stem cells to those neural progenitor cells we talked about, those vital precursor cells, all the way through to immature neurons. And crucially, many of these cells were actually observed in the division phase. That directly confirms active proliferation and growth. They caught them in the act, so to speak.
That is incredible. And importantly, they specified the precise location too, right? Where was this happening?
Yes, absolutely critical detail. These newly formed cells were consistently found in the dentate gyrus. That's a very specific area within the hippocampus.
Okay.
And as you mentioned earlier, the hippocampus is crucial for memory formation, learning, cognitive flexibility. The dentate gyrus is particularly important. It acts as the primary gateway for information flowing into the hippocampus. So finding new neuron formation happening specifically there, that's profound.
Wow.
It confirms an ongoing process of new neuron formation, and it seems to be organized along a continuous neurogenic trajectory. Very similar, actually, to what's seen in other mammals like mice, pigs, and rhesus monkeys.
So it's not just some random biological curiosity.
Not at all. It fundamentally reshapes our understanding of brain plasticity. It shows that the very hardware of our memory center is potentially continually being refreshed and developed throughout life.
So it's a conserved process then, appearing across different species. But were there any unique features about human adult neurogenesis that stood out in this study?
That's a great question. If we connect this to the bigger picture, yes, the neural progenitor cells in adult humans are indeed transcriptionally similar. Their gene activity profiles look alike compared to those found in other species like mice, pigs, and macaques. And they even look similar to human cells during childhood development. So that really suggests a fundamental conserved process across species and throughout our development. However, there were some nuances, some subtle differences. They observed slight variations in which specific genes were active. And interestingly, they also identified some genes that appeared to be specific to adult human cells.
Oh, like which ones?
Two examples they mention are apolipoprotein L domain containing 1, or APLD1 for short, and ribonucleotide reductase regulatory subunit M2, or RRM2. Now, these genes could be unique regulators or maybe structural components involved specifically in human adult neurogenesis. And it's exciting because it potentially offers entirely new targets for future therapeutic interventions, ones tailored specifically to human biology.
That makes sense. Now, what's truly striking, and something I think you might find particularly relevant, is the huge variation they observed between individuals. Some adult humans, despite their age, apparently had many neural progenitor cells, while others had hardly any at all. Does the study offer any, maybe, preliminary thoughts on why this variation exists? Could be genetics, lifestyle, environment, maybe even past experiences, like that epilepsy example they mentioned?
That's a really critical observation, and honestly, it opens up a huge area for future research. The study does point out, as you said, that one 40-year-old subject with a history of epilepsy showed a notably higher number of these progenitor cells. But interestingly, a 58-year-old individual with no known pathology also had remarkably high numbers.
So it's not just disease-related necessarily?
Exactly. It suggests that both biological factors, which could certainly include genetic predispositions and potentially even disease states or other life experiences, might influence neurogenesis in very individual ways. We're clearly not all the same in this respect.
And what about those cases where they found the immature neurons, the baby neurons, but the progenitor cells, the parent cells, seem to be absent? What does that tell us?
Yeah, the study did note that while these progenitor cells were found in most subjects, they were absent in some adolescents and adults. Yet, even in some of those cases, immature granule neurons were still identified. This points to some really complex biological and possibly technical variabilities. It could mean there are other mechanisms for neuronal maturation at play that we don't fully understand yet. Or simply that these progenitor cell populations are just incredibly rare and difficult to capture reliably across all samples, even with these advanced techniques.
Right.
It really highlights the vast individual differences in our brains and just how much more there is still to learn about the specifics.
Okay, so this discovery gives us an incredibly important piece of the puzzle. Understanding how the human brain works and changes throughout life, it really solidifies the idea of brain plasticity, continuous adaptation, proving the brain stays dynamic, capable of producing new cells, even well into adulthood. But beyond just understanding, what are the broader implications here? Why does this really matter for the future, especially for you listening?
Well, this research isn't just academic, not by a long shot. It has potentially profound implications for the future of brain health and possible treatments. Think about it. If we can definitively confirm that new neurons are being formed, even if it's in smaller numbers in some people, it means the brain has an intrinsic capacity for self-repair and adaptation that we might be able to harness.
Imagine the possibilities for regenerative treatments. I mean, this could potentially mean stimulating new neuron formation to address conditions that currently have very limited options, things like certain neurodegenerative diseases, perhaps, or even psychiatric disorders.
That's the hope. And even a small number of newly generated neurons could potentially have a significant functional impact on the hippocampus. That's partly because these immature granule neurons, these new cells, seem to have a period of enhanced plasticity. They're highly responsive to excitation.
Like blank slates.
Sort of, yeah. Incredibly receptive to new information and forming new connections. More easily integrated into existing circuits, perhaps.
That makes sense.
So, yes, this capacity suggests potential avenues for interventions, maybe through pharmacological approaches, drugs that boost neurogenesis, or even behavioral and lifestyle modifications. Things we might do ourselves to enhance this process where it might be faltering. The more we understand the specific mechanisms and triggers for this process in humans, the closer we might get to unlocking entirely new therapeutic approaches for a healthier future. It really brings a new dimension to how we think about resilience and recovery in the brain.
It really does. But while it's groundbreaking, it's also important, as always in science, to acknowledge the limitations, right? What were some of the challenges or maybe the open questions that this research highlights?
Absolutely. Science is always incremental. And yes, drawing firm conclusions about the exact number of neural progenitor cells at different adult ages is still tricky, for example. This is partly due to variations in the nuclear enrichment protocols used across the different samples they studied.
Can you explain that a bit? Enrichment protocols?
Sure. Because these cells are so rare, you can't just analyze a random chunk of brain tissue easily. You need methods to enrich your sample, basically, to concentrate these rare cells or their nuclei before you analyze them. Imagine searching for a few specific rare grades of sand on an entire beach. These enrichment techniques are like using a specialized sieve or magnet that helps you collect more of those specific grains, making them easier to find and study. But different labs or biobanks might use slightly different sieves. making direct comparisons of the exact quantities across all samples difficult. They needed large numbers of nuclei and these specialized enrichment techniques, like sorting for cells that have proliferation markers, biochemical flags, indicating they are actively dividing just to find these cells in the first place.
So even with all these advanced methods, they are dealing with very, very rare cells, which makes precise quantification tough.
Extremely rare, yes.
And this study, it certainly opens many doors, but it also raises new questions, doesn't it? For instance, you mentioned that intriguing observation about the individual with epilepsy having more progenitor cells. Can the study definitively link pathology and neurogenesis at this stage?
No, not yet. That's an important caveat. This particular study wasn't specifically designed or statistically powered, as we say, to draw definitive conclusions about a direct relationship between specific pathologies like epilepsy and neurogenesis. Those were interesting observations in specific individuals, but you'd need dedicated, much larger studies focused specifically on disease cohorts to establish a causal link.
So it's an exciting lead, but not a firm conclusion from this paper.
That's exactly a very important area for future work.
And are there still unknowns about the how of adult neurogenesis, the mechanisms?
Definitely. It's also still unclear if this specific pathway, the generation of immature granule neurons by these proliferating progenitors, is the main or maybe even the sole mechanism responsible for all adult neurogenesis we might observe, or how it might complement other potential pathways of neuronal maturation or plasticity. The brain is incredibly complex. There could be other ways new cells or connections are formed or strengthened that contribute. And as we discussed earlier, that observed individual variation also warrants much, much further study to understand its underlying causes. Is it primarily genetic? Is it lifestyle factors, diet, exercise, stress, environment? Is it a combination of all of the above? That's really the next frontier.
So quite a journey, from a long-standing debate full of conflicting results to this really compelling new evidence. We've unpacked the incredible finding that your adult brain is indeed capable of producing new neurons right there in the memory center, the hippocampus. This deep dive into the human hippocampus really reminds us of the profound, ongoing adaptability of our own minds. And it makes you think, doesn't it? Consider this. If our brains are continuously generating new cells, even into adulthood, what does that mean for how we approach learning, memory, even our mental well-being throughout our entire lives? How might understanding this intrinsic capacity change the way we think about aging or brain health, or maybe even recovery from neurological challenges? It even makes you wonder, what small steps could you maybe take to support this remarkable plasticity in your own brain?
Absolutely. This research truly underscores that the brain is not some static organ fixed after childhood. It's a dynamic, ever-changing landscape. And the more we understand these fundamental processes, these mechanisms of change, the closer we get to unlocking entirely new therapeutic approaches for a healthier future. And maybe just a deeper appreciation of our own incredible biological capacity for adaptation and renewal.
Thank you so much for diving deep with us today and unpacking all of that. Until next time, keep exploring, keep learning, and keep those neurons firing.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.